Here, the good medium must be sufficiently firm and dense to hold the cuttings, and seeds in place during rooting or germination. Its volume must be fairly constant when it is either wet or dry. Any excessive shrinkage after drying is desirable. It must be sufficient retentive of moisture so that very frequent watering is not required. It must be sufficiently porous, means the medium must be sufficiently as like a porous so that the excess water drains away quickly to facilitate adequate aeration. It must be relatively free from weed seeds, nematodes, and various other noxious microbes. It must be a suitable pH. It must contain the suitable pH. The pH mm, measure of the pH relatively acidic or alkaline required for the plant growth. It should withstand steam sterilization without any deleterious effects. How to prepare the media? There are three different types of media preparations, and media mixtures commonly used here is one is soil and its composition, sand and its composition, farmyard manure and its composition. When if we will see this soil and its composition, it is composed of material in solid, liquid, and gaseous state. For better growth of the plant, these are to be in a proper position, proper portions. The texture of soil refers to the relative portion of sand that the particle diameter should be like 1 to 0.5 diameter mm, and the silt of 0.05 to 0.002 mm in particle diameter. Clay should be less than 0.02 mm in particle diameter. The principal textured classes are sand, sandy loam, silt loam, clay loam, and clay. A typical sandy loam may consist of 75% of the sand and 14% of the silt and 11% of the clay. Soil structure refers to the arrangement of the above particles in entire soil mass. And coming to the sand and its composition, it consists of small rock grains from about 1 mm to 0.5 mm in diameter, formed as the result of weathering. Means it forms after the weathering of the rock. Uh, the various rocks, its mineral composition depending upon the type of rock, mainly type of rock, like quartz sand. The quartz sand consisting of silica complex is generally used for the propagation purposes. And what is farmyard manure and how we will prepare the farmyard composition? It is nothing but manure produced in farm having animal excreta, sorry, animal excreta, litter or bedding, and the household material and miscellaneous materials. The farmyard manure should be used when it is well decomposed. The manual value of this manure, uh, though not very high, helps crop production. In many other ways, it adds organic matter to the soil. It promotes crumb formation in soil, leading to the improvement in the circulation of the air. Aeration will be there. Porous material, since it is a porous material, and water in the soils. It makes the soil porous and also increases in water holding capacity as like humus. When coming to the nursery, after media preparation, we have to focus on the nursery. The scale of mulberry plantations and their utilizations is determined according to the plan of silkworm rearing. In order to, yesterday we also discussed about this point. In order to harvest a plentiful of good quality leaves, one requires to adopt good cultivation practices. This generally depends upon the number of plants available in the field, nutrients available in soil, irrigation facility, and drainage, etc. Besides aerial environmental factors, thus it necessary for a farmer to see that all the plants in the field, whether it is growing properly, since they are planted evenly, so we have to look after that. These should not be have any gaps in the field, which causes uneven distribution of the nutrients among the plants that reflects on the quality of the leaf. As yesterday we discussed, we are focusing on the mulberry plant, but our main focus is the yield of the leaf and the yield and quality of the leaf. So the establishment of the optimum plant population is essential to get maximum yield. However, the yield per unit is increased due to efficient utilization of growth factors. Besides all these factors, plant survival factors are essential that plays the major role, especially for the rooting. This problem arises when mulberry cuttings are planted directly in the field, which can be solved by the adopting nurseries. When coming to the bed preparation, nursery facilities the plant or seed for luxuriant growth by supplying essential nutrients like water, soil. Nursery has to be established 4 months before the mulberry cultivation. It is better for the seedlings. Generally, cultivation starts with onset of monsoons. Instead of planting mulberry cuttings directly in the field, it is better to plant seedlings that are grown in a nursery because these seedlings are grown in healthy conditions. These seedlings that are grown in nursery because these seedlings are grown in healthy conditions and thrive better for transplantation. First, a suitable place in the farm is to be selected for nursery. This is plugged deeply with the help of tractor and left for 10 to 15 days after plugging. Latter, plugging in the should be like a crisscross to make the soil to find tilth. The plot is divided into 18 to 10 yards nursery beds with 18 to 10 inches bund. Irrigation provision is made with 30 to 45 centimeters size canal. For each bed, add 20 kilograms of cow dung and mix it well. If the nursery soil is clay, a sufficient quantity of sand is added or the bed with more sand is added with tank silt. Such nursery beds are 7 to 8 inches height to the ground level. How are the planting and maintenance? Before going to this, here I want to show you how to maintain the sand. See here. Here you can see the burnt rice husk and the sand and the soil. Uh, Here just we discussed the cuttings. Here we discussed about the suitable tank, the place in the farm, and selected for the nursery. If you can see here, here you can see the cuttings and the plantation. The distance between the two plantations here you can see. And just now we discussed about the burnt rice husk and sand and soil regions. Here clearly you can see that. And plant how we have to plant and maintain the mulberry plants. The planting distance has just we discussed 15 centimeters between rows and 10 centimeters between plant. And cuttings is followed. Here you can see that 15 centimeters between the rows and 10 centimeters in between the plant cuttings. Plant the cuttings are at an angle with at least one bud at the top, and cuttings must be planted the right way up. The cuttings are given optimum moisture by supplying necessary water. Here we can easily supply the water here because there will be a distance in between two plants, no? So already our soil is porated. That's why if we will apply the water or the nutrients, definitely it will absorb. And the cuttings are given optimum moisture by supplying necessary water. Depending on the water requirements, the beds are irrigated once in a 4 to 6 days. As in the before classes, we discussed that it needs less amount of the water, so that we have to irrigate the beds once soon after we planted, about 4 to 6 days. The cuttings once planted in the nursery are frosted for the several months until they are transplanted. The suitable time for the planting the cuttings is beginning of the rainy season, and the weeding is carried once after 30 to 40 days, and again after 50 days we have to check that whether the weeds are there or not. Intercultivation process is conducted with utmost care not to damage the roots. The cutting sprout within the 5 to 6 days. Nursery saplings are supplied with fertilizer after 5 to 6 weeks, for which complex fertilizer, the ratio of the NPK, nitrogen, potassium, and phosphorus, nitrogen, phosphorus, and potassium ratio should be 25 east, 25 east, and 25 east. Means equal amount of 1 is to 1 is to 1 ratio per 1 kg per hectare. That is the advisable concentration of the complex fertilizer NPK. Nursery is screened from sun with a bamboo mat to prevent cuttings from the desiccation. Planting material. Cuttings are obtained from the lower part of the thick shoot, which results in vigorous rooting. The shoots are prepared from a selected variety. The shoots of 6 to 8 months old, dark brown in color, and pencil thick is chosen for cutting. Cuttings of 18 to 15 centimeters along with 3 to 4 buds are required for irrigated mulberry. Rain-fed mulberry requires 22 to 24 centimeters along with 5 to 6 buds. The ends must be 45-degree angle. We have to cut the ends in the 45-degree angle without bark peeling and split. Spacing. It favors equal distribution of the soil nutrients. You can see here in the figure C, and just now we discussed about the cuttings. Here you can see the axillary buds and how to uh, make the 45-degree angle for irrigated and for rain fed. And just now we are discussing about the spacing. In the image C, you can see the nursery after plantation. A thick population induces competition for nutrients, soil moisture, and minerals, etc. Among the plants, uneven or irregular spacing results in irregular growth and poor crop results. If spacing is less, it obstructs the evaporation and increases humidity as the time passes. High humidity induces incidence of powdery mildew. Further, deficiency diseases are also appear The spacing of 15 centimeters between the rows and 10 centimeters between the cuttings is suitable, which is sufficient till it attains 5 to 6 weeks age and becomes ready for the transplantation. How we will do the transplantation? Here it is a cultivation practice. Transplantation is a cultivation practice in which saplings or seedlings are uprooted from nursery and planted in the field. Saplings grow to a height of 3 to 5 feet with 4 months within the 4 months period. Nursery is given a lot of water for 2 to 3 days before uprooting. It is the uprooting is to loosen the soil and not to cause any damage to root system while uprooted with uh, sickle. Then these are bundled carefully and wrapped in a wet gummy bags for transporting. Then this is ready for transplantation. When coming to the propagation through seeds, here you can see the image, the stages of seed germination in mulberry, propagation, A, B, C, D, and E. Propagation. Mulberry is propagated through seeds, which are very easy and cheap. This process brings about a varied population for selection and hybridization. The seeds are used for obtaining stock material for grafting. Seed collection. Seed collection collected from the ripe fruits in the month of March to April in tropical regions and May to June in temperate regions. There is no dormancy in mulberry seed but lose viability with the passage of time. If you will store the seeds, that will lose their seed viability. If we preserved beyond 3 months, the seed should stored in a sealed airtight container that should kept in a cool place. However, freshly harvested seeds show high germination. The ripe fruits are collected in separate containers filled with water and squeezed to separate pulp from the seeds. The floating seeds are removed along with water and the heavier seeds settle at the bottom of the water. Heavier seeds are collected carefully and dried on a blotting paper. The seeds are small and uh, and the amount is 3000 per 1 liter. And coming to the nursery plots, the plants grown from seed is called seedling, which is grown in nursery. The nursery plot is selected preferably under shade in the farm. The soil is thoroughly dug to prepare to a fine tilt. The sowing medium is prepared by mixturing equal volume of the red earth, sand, and FYM as just we discussed, and nursery beds of 0.9 m size, 9 meter size are prepared with the provision to protect young seedlings from excessive light and temperature. Sowing of seeds. Seeds are soaked for a day in water to remove the dormancy and for making the seeds into soften. This is only the easiest method and successful germination method. And seeds are broadcast or sown not more than 2.5 centimeters deep in bed using a rope. Uh, with the help of the rope, we can measure in straight line a distance of 2 to 3 centimeters between rows and 1 centimeter between seeds is advisable for better germination. After sowing, bed is covered with soft soil and applied water gently. We have to apply the water gently after the sowing. Seed germination. Environmental conditions are the vital plays the vital role, and beds are covered with a shade made up of bamboo stripes or palm or coconut leaves to protect from the hot sun. These mats are placed 1 foot above the nursery beds on pegs fixed around. Low temperature and colored light delays seed germination. Seedlings, these require proper nutrition for uniform growth. The seedlings of 3.5 to 5 centimeters height are picked from dense areas and planted in thin areas. During cool hours of the day, on the cloudy days, direct sunlight is allowed to fall on seedlings to enhance plant growth. After 3 months of age, seedlings are transplanted with a distance of 22 centimeters between the plants. These can be either used for grafting, stock plant, or allowed to grow for 2 years to raise tree plantation. Propagation through cuttings. Here you can see the structure. How can we propagate the mulberry plant? Mulberry plant through the stem cuttings. Here you can see in the figures. Most of the tropical varieties of mulberry can be easily propagated on large scale through cuttings. It is the easiest way and common, and it is a very popular method in the India. But this method is restricted to acclimatized in the local varieties. The plants are selected on the basis of nutritious leaf and high yield and quick growth resistance to the diseases. And also insect and pest drought resistant varieties. This propagation is very cheap and easy and quick. Mulberry being a insect pest and dough resistant varieties, this can be cross pollinated. The improved traits of the cultivar are retained by this method. Shoots of proper maturity within 8 to 10 months old with active and well developed buds are selected. Over mature, the tender tips are rejected. The branch must be 10 to 12 millimeter in diameter. The cuttings of 18 to 20 centimeters along with 3 or 4 active buds is prepared with a sharp implement so that neither the bark nor the wood is damaged. The ends must be slant as we discussed in the previous sessions. The slant must be 45-degree in angle without damaging the bark or uh, split. For irrigated farm, cutting size 18 to 20 centimeter long with 3 to 4 buds. For a rain-fed farm, cutting size 18 to 20 centimeters along with 5 to 6 buds. Development of the roots. The roots develop from the basal end of the, here you can see the basal end. This is the basal end and here it is the root development. Basal end of the cuttings which is near to the soil. The roots developed endogenously. The root initials develop internally, finally produce the root primordium. so as to form root tip which grows outward and emerges at the right angles to the stem. Simultaneously, callus tissues develop at the basal end of the cuttings and becomes stem of the future plants. Here you can see the formation of the callus at the cutting the cut end. Here it is the callus. Propagation through grafting. How we can propagate this plant through the graft. See these pictures A, B, C, D. The first one is what is propagation through grafting? Propagation through grafting, it is the technique of joining a branch of one plant into a rooted plant in such a way that they united with a organic union formed in between the plants and finally grow as one plant. It is practiced where the plant cannot be propagated through cuttings because of poor rooting. First one is scion and another one is stalk. What is scion? The part of the graft combination which is become the upper portion of the shoot system of the new plant. What is stalk? The part which is become lower portion or the root of new plant to supply nutrients. The stalk must be a local one, handy variety, and scion is selected from the desired variety. The total strength of the future plant variety depends on the stalk, as it alone influences the growth. Thus, the stalk should be in a more advanced stage of growth than the scion. This method facilitates to get desirable qualities which cannot be propagated by other means. What are the basic principles of grafting? The basic principle of the grafting is compatibility of a stalk and scion. And the next one is maximum cambial contact between the scion and stalk. And the other one is local availability of stalk and the superiority of scion. Selection of plants. The stalk should be from 1 to 2 years old seedling. It should be healthy and highly resistant to diseases and pests. With the rich root growth, stalk should be slightly bigger and the same size as that of scion. Scions are chosen from 1 year old plants and only middle part of the shoots are used, leaving the top and the base regions. The leaves from these plants should not have been plucked during the previous year. Further, it should have narrow and compact pith. How to prepare the formation of graft union? First one is establishment of contact of large area of cambial region of both stalk and scion. As we discussed, scion means the root system of the new plant and stalk means scion means shoot system of the new plant. Stalk means root system of the new plants, means lower portion. Stalk is lower portion. Scion means upper portion. Don't be confused with that. So how to the formation of graft union? So this establishment of the contact of a large area of cambial region of both the stalk and scion and stem and root. Production and interlocking of parenchymal cells, that is callus tissue of both the stalk and scion. Differentiation of new cambia across the claw bridge. Formation of xylem and phloem from the new vesicular cambium in the callus bridge. The intercession of the scion into the stalk establishes the organic union by the formation of secondary vesicular tissue. The cell division increases to form callus tissue during the proper temperature between 20 to 40 degrees. Further, it is necessary to maintain the high humidity. Depending upon the type of material used, and also its nature, age, and the portion of the stalk, graftings are root grafting, whip and tongue grafting, and approach grafting. There are different types which is used for the formation of the graft union. In mulberry only, root grafting is practiced. When going to the root grafting, it has two methods. The root grafting has two methods. One is in situ root grafting and simple root grafting. What is in situ method? The scion is grafted on the root of the existing plant without disturbing the root from its original place. Though a more vigorous plant is obtained, in this case, only one graft is developed from a seedling. In simple root grafting, the roots from a good rooting variety are removed, cut into convenient pieces, and used for grafting by inserting the scion into the root piece. And planting the graft in a nursery, it is more economical for multiplication as more grafts can be prepared from the root of the plant. Here you can see the first picture, figure A, root grafting, figure B, stem, and figure C with 8 to 10 centimeters length here we are showing, and figure D, here we are grafting both the root part and the shoot part. And propagation through budding. Budding is also a method for grafting where in only a single vegetative bud with a piece of bark is used as a scion material. As we know, scion material means upper part. That is why it is also called as bud grafting and has all the advantages of the grafting. Budding is practiced as a means of vegetative propagation in mulberry, particularly in temperate regions of the country. It is practiced whenever scion material is scarce, when the scarcity of the scion material is there and involving large scion material causes disease transmission. Depending upon the types of bud, there are various types of the budding process. One is tea budding, that is also called as shield budding, inverted budding, that is inverted tea budding, patch budding, flute budding, chip budding, and ring budding. What is tea budding? Check this picture for tea budding. It indicates, the tea budding is indicates, here you can see the 4.5 figure for tea budding. It indicates a cut given on the stalk. It is the shape of letter, it looks like a shape of letter T, which is used for mulberry. A cut is given on the stalk 1.2 centimeters in diameter at the internodal region with the 25 centimeters height from the ground level. A vertical cut is given to the length about 1.2 centimeters, and then cut horizontally at the top of the vertical cut, which appears like a T shape. Then slowly bark is removed to insert the bud. After, you can see here clearly the measurement of the cut around the bud and the removing of the cut, and how to place the bud. This bud is ready for insertion, and bud inserted and wrapped, and shoot growth from the bud in about an year. So here what we did, after selecting the required bud, make a slanting cut in the bud and lift it upwards about 2.5 centimeters length above the bud. Gently remove the bud and insert in T-cut of the stalk and wrap the union tightly with a polythene stripe exposing only the bud. When coming to the inverted T-budding, inverted T-budding, here you can see the inverted T-budding. It is just exactly opposite to the T-budding. In T-budding, there is a possibility of water drops entering the cut made on the stalk and may lead to damage the bud inside. In inverted T method, water entry is prevented because the shoot with the bud, here you can see, and remove the bark with the bud at the place of root scion. Here we can insert the bud invertedly and flute the bud with the help of the polythene cover. That is finished by wrapping. That will not allow any water enters into the bud for damage. What is patch budding? Patch budding, here you can see the structure of the patch budding, levels of the patch budding. In this patch of the bark, with an active bud is removed from the bark of the scion, a patch of bark is similar in size and carefully removed from the stalk in the internodal region. Then patch with bud of scion is inserted on the stalk and properly bandaged with the soft fiber and covered with grafting wax or clay for further developmental stages. Here you can see the patch budding, the selected stem and a shield bud, making the T-shaped cut on the stalk plant and inserted bud in the T slot, and the bud is wrapped and wrapped with the help of soft fiber and covered with grafting wax or clay. And next one is flute budding. The flute budding method is a modified method of patch budding method. The bark, that is stalk of the plant, is cut to a length of 2.5 to 3.5 centimeter round and removed as a sheath at the internodal region. The removed bark looks like a C letter. The bud of the scion is also removed in similar manner and inserted in the stalk and bandaged properly after applying grafting wax. Here you can see the figure of flute budding. Here the stem with the bud and the bud and the shade. Here the cut of the inverted T cut on the stalk, and here the inserting of the bud, and once the bud is inserted, complete the bud grafting after tying the rubber band, and also applying the grafting wax for further germination process. So for this we have to take some precautions. What are the precautions here we have to take? Precautions. The size of the scion and the cut portion of the stalk must be always equal. Otherwise it will not going to be fit. While removing the bud, no woody portion should stick to the bark. It is necessary to remove all the buds below and the above budding portion only. There should not be an empty space in between the scion and the cut portion on the stalk. The stalk should not be damaged. The cut or site of the insertion should be bandaged properly to prevent entry of the water and air. It is necessary to inhibit sprouting of other buds on the stalk. These are the precautions. And when coming to the propagation through layering, so there is a process called layering for the propagation. What is that? It is a process of development of the roots on a stem while it is still attached to the parent plant. When the plant is already attached to the parent plant, we'll do this method for developing the roots on the stem. The rooted stem is detached to grow as a new plant and which can be called as a layer. Root formation on the stem during layering is stimulated by various treatments which cause the interruption in downward translocation of organic materials like carbohydrates, auxins, and other growth regulators from the leaves and the shoot tips. These materials accumulated near the point of the treatment and caused the formation of the callus, from which roots are developed. This method is simple, safe, and cheap than any other method. This method favors to get a large sized plant in a short time. There is no chance of root drying. The main disadvantage of this method is it is a high time consuming method and also very expensive and not suit for large scale production. After root development, the branch is separated from mother plant and used as a sapling. This method helps to fill gaps in the fields. There are other types of, there are the types of layering, like three types of layering. One is simple layering. Wait, here we will see the structure. Here we will see the figure. Now we are talking about the simple layering. In this, a lower branch, here you can see the lower branch, that bent and bark is removed to a length of 2.5 to 3 centimeters in middle region of the branch. The branch is then bent slowly to the ground and treated part is covered with soil, leaving the terminal end freely and exposed. It is protected from dislocation by inserting wooden pegs or fork. Root initiation takes place at the bent and buried end. Within 2 or 4 months, which is then cut off from the mother plant and transplanted to grow individually. From here you can take a new plant, and from here you can take a new plant. When coming to the trench layering, here you can see the trench layering. This is a branch of growing plant is bent and covered with the soil and manure, leaving the tip exposed to the roots developed from the buds along with the stem grows upwards to the soil and roots on opposite direction. This process is better to get number of layers from a single branch. This one here you can see the ground layering trench. From one plant about 4 plants are coming. How much place if the root is occupies, that many plants will come in the trench layering. It depending upon the branch, how it is bending, how it is moving. Air layering. Here air layering. This is the method is to induce roots on a aerial shoot. Here aerial shoot. The method involves making a girdle at the point of 20 to 40 centimeters from the tip of shoot just below the node by removing a stripe of bark 2 to 5 centimeters. Exposed bark is scraped to remove the traces of phloem or cambium. The girdle is covered with the propagating medium and tied with a polythene stripe not to allow water or insect into the cutting portion. After 2 to 3 months, polythene cover is removed and the rooted stem is cut for plantation. Here you can see. This is about the layering. Is a part of propagation through layering. When coming to the nutritive values of the mulberry, the silkworm eats only mulberry leaves to make its cocoon, producing silk. Mulberry leaves are rich in protein and amino acids. It is known that there is a high correlation between the leaf protein level and production efficiency of the cocoon shell, which means the cocoon shell weight to the total amount of the mulberry leaves consumed by the silkworm. Therefore, an increase in the protein level of the mulberry leaves may lead to improvement in the cocoon productivity. What is the chemical composition of the leaves? Here the chemical composition of the leaf and leaf variety and the maturity is depending upon the chemical composition. However, the basis of analysis carried out by the CSR and TI Mysore, the chemical composition of the leaf is like the moisture content, which ranges from 65 to 78 percentage in the mulberry leaf. And when coming to the protein content in the leaf of the mulberry is 19 to 25 percentage, and the mineral presentation 10 to 15 percentage, and reducing sugars are presented about 1.2 to 1.9 percentage, and the sugars about 10 to 15 percentage are presented in the nutritive value of the mulberry, the leaf component of the mulberry, which is this analysis is given by CSR and TI Mysore. It is a known fact that the quality of mulberry leaf improves growth and development of silkworm. Internally, Silkworms indicate mulberry silkworms because most of the available silk is from mulberry. This is an advantage to the farmers to rear silkworm in their houses. These worms are Bombax mori, eat only mulberry as there is no alternate for the mulberry. Synthetic dyed production and its effects on the health of the silkworms did not show complete results. Thus, silkworm rearing requires the quality of the leaf, the quality of mulberry leaf depending upon the environmental factors, cultivational methods, irrigation, soil structure, soil type, pruning methods, and fertilizers and rainfall factors. These are all the factors which makes the major role for the quality of the leaf. Leaves will also be a succulent and tasty for eating for the silkworms. Silkworms eat these leaves and increase their body and produce a good quality of the cocoons. Mulberry grown in only loamy soil will have high values of water and proteins and carbohydrates. Fibers are very less. Further, leaves mature slowly. Soil with a little sand and pebbles induces early maturity of leaves. These leaves are with less moisture, proteins, and minerals, while carbohydrates fibers are very high. Mulberry grown in these soils are given minerals with fertilizers in equal quantity to increase leaf chemical and physical characteristics. Mulberry grows healthy in good rainfall and irrigated areas. And mulberry grown in these conditions are with high nutrients, smooth and succulent leaves. As we already discussed, mulberry leaf quality is depending upon the physical and chemical properties, especially chemical properties influence the mulberry leaf food values. And also these leaves are with 64 to 83 percentage of water. At the early stage of silkworms require much water, while late age worms require little water content. Thus, water content is adjusted according to the age of silkworms. Proteins are essential for silkworm production. Thus, the ratio of proteins should be more in mulberry leaves. Mulberry leaves are with globulin, glutoline, prolamine, albumin, and protease. And the amino acids like glycol, alanine, valine, leucine, aspartic acid, glutamic acid, phenylalanine, proline, oxyproline, cysteine, cysteine are presented. All these are effective on the health of silkworm. Carbohydrates of leaves are better healthy growth of early age silkworms. Depending on the environmental conditions, carbohydrates are useful for the growth of the leaves and preservation. Thus, a leaf harvest and storage should not affect on the nutrients. Fats are very less in the leaves, but the silkworm body synthesizes high amount of the fats utilizing other compounds, especially carbohydrates, especially calcium, phosphorus, silicon, manganese, and magnesium, iron, copper, and zinc. Among these, calcium and silicon mostly useful for leaf growth. Manganese for chlorophyll, phosphorus, phosphorus for protein formation in the leaves, Further, these materials or elements are for alkalinity of silkworm body. What are the organic substances here we need is makes the delicate parts and comes down when leaves are grown. Mulberry leaves possesses vitamin A, B, C, and the chlorophyll is 0.15 to 0.35% in leaves, which changes according to their growth. Tomorrow we will discuss, in the tomorrow's class, we will discuss about the chemical composition of the mulberry leaves and the nutrients of the mulberry leaves used for the different instars and mulberry breeding, and also ex-situ conservation. Then what are the new varieties evolved, and we will discuss the genetic breeding strategy in the mulberry and for the temperate hills. And here we also discuss about the hybrid vigor in our next class, along with the other topics like cultivation practices. Tomorrow we'll start with cultivation practices. This is about today's class. Hope you enjoyed the class. With this, I'm ending today's class.